Namaste. Myself, Name-Shivi Saxena, Faculty of Physiotherapy and Diagnostics, and my discipline is Medical Laboratory Technology. So, welcome to Digital Version 2.0. And my today topic is Spirochete staining by Fontana's techniques. Okay, so let's start my topic. Firstly, introduction. So, spirochetes are spiral shapes shape gram-negative bacteria, and they are unique in morphology and locomotion. The spirochetes is spiral in shape, so its size is measured by measuring its length and width. Generally, it is 5 to 500 μm in length and 0.1 to 3 μm width. Spirochetes has endoflagella that run through periplasm, and the endoflagella may be 2 to 100 on the number, and these flagella are bound around the helical cells in between protoplast and outer membrane. And these spirochetes are extremely delicate organisms, so some special technique is used to stain them. So there are two techniques. There are two techniques to stain these spiral-shaped organisms. First, Fontana's staining techniques, and second, Becker's staining techniques. Here we are going to study Fontana staining technique in detail. So let's start. Fontana staining technique. Firstly, aim. So to stain spirochete by using Fontana staining techniques. So requirement: first, Fontana's fixative; second, Fontana's mordant; third, Fontana's stain; and fourth, 95% ethanol; and 5, tartar sample. So procedure: take a pin and dip in alcohol and flame sterilize the pin. Second step, this sterile pin is used to take a tartar sample present in interspecies on teeth. So now this tartar sample is mixed in the sterile distilled water, and a drop of this suspension is taken with the help of a sterile wire loop on a clean grease-free slide, and smear is prepared. Okay. Next step, this smear is air dried but not heat fixed. Okay. So this smear is fixed by using a chemical fixative that is Fontana's fixative. This slide is placed on a coliform jar containing Fontana's fixative for 3 times at the interval of 1 minute. Okay. And next, further the fixative is washed off and the slide is treated with 95% alcohol for 3 minutes. Okay. And then after 3 minutes, the excess alcohol is washed off, and the slide is held with the help of forceps and passed through a flame to remove excess alcohol. Okay. And in the next step, Fontana's mordant is added on smear and heated till steam appears. Okay. And after steam appears, the slide is washed with water and air dried. Okay. And next step, this air dried slide is now treated with Fontana's stain and heated till steam rise. Okay. After steam rise, the slide is washed with distilled water and air dried the observed under oil immersion. Okay. So mechanisms of Fontana's stain. So The spirochetes is very delicate organism. This easily get destroyed by heat fixation. So here heat fixation step is avoided. Okay, and hair smear is treated by fontana fixative, and this fixative contains a chemical like formalin and glycerol acetic acid. Okay, and after fixation, the smear is treated to Fontana's mordant, and this mordant contains tannic acid, which increase the affinity of the stain toward stain. And further, the stain is treated with alcohol and Fontana stain. And Fontana stain contain ammonical silver nitrate, and when this stain is heated, it forms silver oxide is formed, and this silver oxide precipitate on the organism and increase the diameter of the cells and stain the cell wall. So result, after completion of Fontana's staining procedure, the spirochetes are stay brownish black in color. Okay, so this session is completed, and any query in mention in comments box, and thank you so much. Namaste. Namaste, myself Name-Shivi Saxena, Faculty of Physiotherapy and Diagnostics, and my discipline is Medical Laboratory Technology. Welcome in digital version 2.0. My today topic is gram staining. So let's start my topic. Firstly, introduction. Gram staining is the common, important, and most used differential staining techniques in microbiology, which was introduced by Danish bacteriologist Hans Christian Gram in 1884. This test differentiates the bacteria into gram-positive and gram-negative bacteria, which help in the classification and differentiation of microorganisms. Principle of gram staining. When the bacteria is stained with primary stain crystal violet and fixed by the mordant, some of the bacteria are able to retain the primary stain and some are decolorized by alcohol. The cell walls of gram-positive bacteria have a thick layer of protein sugar complex called peptidoglycan and lipid content in blue. Decolorizing the cell causes this thick cell wall to dehydrate and sink, which closes the pores in the cell wall and prevents the stain from exit the cells. So the ethanol cannot remove the crystal violet iodine complex that is bound to the thick layer of peptidoglycan of gram-positive bacteria and appears blue or purple in color. In case of gram-negative bacteria, cell wall also takes up the CV iodine complex, but due to the thin layer of peptidoglycan and thick outer layer which the form of lipid, iodine complex get washed off. When they are exposed to alcohol, decolorizer dissolve the lipids in the cell wall, which allow the crystal violet iodine complex to leach out of the cells. Then when again stained with safranin, they take the stain and appears red in color. Reagent used in gram stain. First, crystal violet, the primary stain, iodine, the mordant, and a decolorizer made of acetone and alcohol. Alcohol is 95%, and fourth and last, safranin, the counter stain. So procedure of gram staining. Take a clean grease slide and prepare the smear of suspension on the clean slide with a loop full of sample. Next, air dry and heat fix. Firstly, crystal violet was poured and kept for about 30 seconds to 1 minute and washed with water. And fluid the grams iodine for 1 minute and wash with water. Then wash with 95% alcohol or acetone for about 10 to 20 seconds and wash with water. Last, add saffron for about 1 minute and wash with water. And air dry, blow dry and observe under microscope. So interpretation, gram-positive bacteria show blue and purple color and gram-negative bacteria show red color. So examples, gram-positive bacteria, Actinomycetes, Bacillus, Clostridium, Corynebacterium, Enterococcus, and Lactobacilli, Bacillus, Mycoplasma, and Streptococcus, Staphylococcus, etc. And gram-negative bacteria, E. coli, Salmonella, Shigella, and other Enterobacteriaceae, Pseudomonas, and Helicobacters, and acetic acid bacteria, and etc. So this session is complete, and any query in mention comments box. And thank you so much. Namaste. Namaste. Myself Name-Shivi Saxena, Faculty of Physiotherapy and Diagnostics, and my discipline is Medical Laboratory Technology. So welcome in digital version 2.0. So today my topic is acid-fast stand. So let's start my topic. Firstly, introduction. It is the differential staining technique which was first developed by Giles and later on modified it by Nielsen. So this method is also called Giles-Nielsen staining techniques. Nielsen is 1883 used Giles Carver fusion and heat then decolorized with acid alcohol and counter stain with methylene blue. This Giles-Nielsen staining techniques was developed. The main aim of this staining is to differentiate bacteria into acid-fast group and non-acid-fast groups. So this method is used for those microorganisms which are not staining by simple or gram staining methods. Particularly, the member of genus Mycobacterium are resistant and can only be visualized by acid-fast stain. So, principle of acid-fast stain. When a smear is stained with Carver fusion, is solubilized, the lipodial material present in the bacterial cell wall. But by the application of heat, Carver fusion further penetrates through lipoidal wall and enter into cytoplasm. Then, after all cell appears dead, then the smear is decolorized with decolorizing agent, that means 3% HCl in 95% alcohol. So, but the acid-fast cells are resistant due to the presence of large amount of lipodial material in their cell wall, which prevent the penetration of decolorizing solution. And the non-acid-fast organisms lack the lipodial material in their cell wall due to which they are easily decolorized, leaving the cell colorless. Okay, so then the smear is stained with the counter stain methylene blue. Only decolorized cells absorb the counter stain and take its color and appears blue while acid-fast cells retain the red color. Summary of acid-fast stain. So firstly, application, primary dye, reagent Carver fusion, color, acid-fast red show in red color and non-acid-fast is red color. Okay, and application decolorizer, reagent acid alcohol, and color, cell color, acid-fast red and non-acid-fast colorless. Next, application counter stain, reagent methylene blue, and cell color, acid-fast show in red color and non-acid-fast is blue color. Okay, so let's start procedure of acid-fast stain. So prepare bacterial smear on clean and grease-free slide. Okay, so using sterile techniques. Second, allow smear to air dry and then heat fix. Alcohol, firstly, alcohol fixation. So this is recommended when the smear has not been prepared for sodium hypochlorite, that means bleach, and treated sputum and will not be stained immediately. So Mycobacterium tuberculosis is killed by bleach and during the staining process. So heat fixation of untreated sputum will not kill Mycobacterium tuberculosis, whereas alcohol fixation is beneficial. Okay. Next point, cover the smear with carbon fusion stain. Next step, heat the stain until vapor just begins to rise and do not overheat. Okay, so allow the heated stain to remain on the slide for 5 minutes. So heating a stain, great care must be taken when heating the carbon fusion, especially if staining is carried out over a tray or other container in which highly flammable chemicals have collected from previous staining. And only a small flame should be applied under the slide using an initiated swab previously damped with a few drops of acid alcohol or 70% ethanol or methanol. Do not use a large ethanol swab because this is a fire risk. Okay. Next, wash off the stain with clean water. So note the point, when the tap water is not clean, so wash the smear with filter water or clean boiled rainwater. Okay. Next step, cover the smear with 3% acid alcohol for 5 minutes or until the smear is sufficiently decolorized. So example, pale pink color. And next, wash well with clean water and cover the smear with malachite green stain for 1 to 2 minutes using the longer than when the smear in thin. Okay, next step, wash off the stain with clean water and wipe the back of the slide clean and place it in a drain rack of the smear to air dry. Do not blot dry. Okay. So examine the smear microscopically using the 100x oil immersion object. Okay. So interpretation of acid fast state. So firstly, acid fast show is bright red to intense purple color. Red state or slightly curved rod occurring singly or in small groups may appear bedded. Okay, and non-acid fast bacteria is a blue color. Okay, so examples of acid fast stain. Acid fast stain positive Mycobacterium tuberculosis, Mycobacterium segmentitis, and non-mycobacterial bacteria, nocardia, and Conidian parasites, so Cryptosporidium. Okay, so this session is completed, and any query in mention comment this comments box. And thank you so much. Namaste.